cells. So here, for example, in terms of the first step, recognition after chemotaxis, followed by recognition and adherence. So when we are talking about recognition, recall the previous subtopic 7A. We've discussed about mannose receptor before, right? Scavenger, toll-like receptor, right? So these are the PRRs involved in the recognition and adherence. So this receptor on the surface of the phagocytes will recognize a specific pattern on the surface of the pathogen here, whether PAMs or MAMs. So these are the molecules involved in the process, recognition and adherence. So here the step-by-step process. The first one is chemo extraction by the here C5A, and then the CB coats organism and attach to receptor. So it allows on the surface of the phagocytes. We know that there is C3b receptor, right? So this receptor will be specifically will allow the binding. So the surface of a phagocyte will not just blindly bind to the pathogen. There is some factor that will cause them to bind. So this is a very specific interaction actually. So when you find a specific pattern coated, they will bind, and followed by the engulfment into a structure called phagosome inside the phagocytes. So once the phagocytes get trapped, they need to be digested, right? So there will be another organelles in the phagocytes, which is lysosome, containing a lot of antimicrobial substances, toxic substances against foreign materials, again the microbes. So this lysosome will be fused to the phagosome structure, to the previous vesicle structure as a result of the digestion before. So it will fuse. So what happened? So there will be a lot of antimicrobial attack the bacteria and cause them to be digested into smaller fragment of protein, and lastly will be eliminated from the phagocyte through exocytosis process. So these are basically a detailed process of the phagocytosis. So when we discuss them in context of immune response. So I'm not going to go into details on each receptors of the phagocyte surfaces because we've discussed this before. Here, Fc receptors, C3b receptor, right? Example of receptor could be present on the surface of the phagocytes. So on the surface of the phagocyte, there wouldn't be any one single receptor that could be different that allow them to recognize whether the pathogen has been coated by the opsonine or single bare pathogen that recognize with different molecule on the surface and so on. So that's why they should be having a lot of different receptor on the surface. So normally this kind of immune response, innate immunity, the phagocytosis is very common and they work quite efficiently across different type of bacteria and virus. Only there is certain exception, then this kind of mechanism would be failed. So one very common criteria of the bacteria that will not allow phagocytosis to kill them is through by the capsule formation. So any kind of bacteria that we know normally produce capsule, normally they will not be working, killing them with phagocytosis will not be working. So the immune response need to use another mechanism. So for example, the presence of specific protein on the surface, M protein, will avoid the attachment of the phagocytes. So the capsule layer of the S. pneumoniae, H. influenzae also may cause the bacteria to escape phagocytosis. So these are the details on the formation of the before the digestion occur. Let's look at the mechanism of killing inside the phagolysosome. So what is actually happening in phagolysosomes? So I mentioned just now there are a lot of antibacterial toxic compound inside the lysosome, right? That allow the structure of phagolysosome to digest the pathogens. So what are they actually, the antimicrobial compound? So it may include a lot of free radicals. So one of them, very common one, is the oxygen reactive radicals, reactive oxygen species, the ROS. So how do the ROS is actually produced? Normally, ROS is produced by neutrophil and macrophages that is triggered whenever the enzyme on their membrane, cell membrane that contain NADPH oxidase, phagosome oxidase, activated by the phagocytosis process. So we know that when the phagocytes want to kill the pathogen, they need to engulf, right? So the formation of the engulfment, the pseudopods of the phagosome surrounding the bacteria will actually activate this enzyme, NADPH, phagosome oxidase. So once they are activated, this enzyme will reduce oxygen into oxygen radicals, superoxide anion, which is toxic. So this species of ROS can then convert it into hydrogen peroxides with the interaction of another enzyme here, superoxide dismutase, to produce hydroxyl radicals. So as a result of formation of the initiation of the phagolysosome, the phagocytosis is triggered. It causes certain enzyme on the phagocyte membrane being activated and produce ROS, oxygen radical, as well as hydroxyl radicals. So that is one way how. Meaning originally inside the lysosome, inside the phagosomes, it does not contain the radicals, the free radicals, because these are a very toxic compound, right? So there shouldn't be any cell that carry this toxic compound anywhere in our body, right? Because they are very dangerous. So that's why, because of the initiation of the phagocytosis, only this free radical will be produced. And the other type of killing mechanism could be involving the RNS, nitrogen free radicals, reactive nitrogen species. So this kind of radical species is usually produced when there is an activation of TLR receptor. So it's involved TLR receptor, nitrogen species will be produced. Nitrogen species also is a very potent agent, and it can combine with the oxygen radicals, and later